Today I'm going to explain the central dogma of molecular biology, which says that DNA can duplicate. You know that the DNA needs to be duplicated during the S phase in the cell cycle before the cell division. And also DNA can transcript into RNA, the messenger RNA, and this messenger RNA can be translated into proteins. This is the central dogma of molecular biology. Here we have a cell, okay, and this is the DNA in black. The RNA, the messenger RNA, is in green, and the transferent RNA is in red, and this is a ribosome. So first of all, I'm going to start explain the duplication of the DNA. How this occurs? You know that the DNA is a double chain, okay, with two strands in which are the nitrogenous bases. Okay. For duplicating it, the first step is to uncoil, okay, desenrollar the double strand, and that is made by an enzyme which is called topoisomerase, okay. That enzyme is going to uncoil the DNA, and then another enzyme which is called helicase, is going to separate the strands. When this is done, we have the replication fork like tenedor, okay? In Spanish es la horquilla de replicación. This occurs in a point which is called the origin of replication, okay? Here is the origin, and the strands are going to be opened in that direction. Now, the second step, or the third step, when we have the replication fork, is that a new enzyme called DNA polymerase, then la ADN polimerasa, is going to create new strand by adding nitrogenous bases. How does the DNA polymerase do this? It reads the DNA from the 3' extreme to the 5' extreme. The direction of the reading of the DNA is from the 3 to 5, okay? 3 to 5 in this direction. In this direction, the DNA polymerase is going to add the complementary bases of these ones. So here is going to add a T, G, the complementary ones, okay? C, A, T, A, C, and C. And in the other strand, the DNA polymerase goes reading from 3 to 5, from this part to this part. And it adds the bases, the complementary bases, which are going to be G, G, T, A, T, G, C, A, A. Now we have here is a double chain or double strands, and here is the other one, okay? We have from a double chain, from an original double chain, we obtain two ones, and this is semi-conservative. We say that this is semi-conservative replication. Why? Because the new double chain is going to have two chains or strands, which are going to be the old one and the new one. And this one which is double, okay, is going to have the old one and the new one. If you check, this was TATAGC, and the new ones are TATAGC, and this TATAGC. You can check that these doubles and these doubles strands are the same as the first ones. And that's how replication occurs. The topoisomerase is going to continue opening the fork, and replication is going to take place. But now, there's a little problem. Replication goes from 3 to 5, okay? And the new strand is going to be, here is the 3 extreme, this one has here the 5 prime extreme, and here the 3 prime extreme, and the same in this one, 5, 3, okay? So, as the fork opens, we are going to find that this strand is going to be created continuously, okay? And we say that this is the leading strand. But this other strand cannot be created continuously because the fork opens in that direction, and the duplication occurs in this direction. So, this strand is the lagging strand, okay? Esta sería la hebra adelantada y esta sería la hebra rezagada o atrasada, ¿vale? Lagging strand. ¿Por qué? Tiene que esperar a que se abra. This strand has to wait for the opening of the replication fork, okay, for being synthesized. I'm going to explain now. Tiene que esperar a que se abra la horquilla para seguir sintetizándose de atrás hacia adelante, okay? So, we start with the double strand, okay? And here the replication fork is opened. Yeah? Here we have the basis and the basis. And as you can see, in this case, here is the 3 prime and the 5, and here is the 5 prime and the 3 prime string. So the replication is going to be in this direction and here in this direction. Okay. Now the topoisomerase continues opening the double chain, okay, the double strand, or for making the fork. And this here, the polymerase is going to continue adding bases here. It adds bases here and it continue adding bases. But in this case, it starts here and it adds bases, but it has to wait for the replication fork opens, okay? When it's open, and now here starts adding new bases, okay? This one synthesize, the strand is going to be synthesized continuously, and in this case it's not going to be continuous because it starts in a place and it synthesizes a part of the chain of the strand, and when the fork is opened, it has to wait, and then it synthesizes another part or another piece of the strand, and then when it opens, as the replication fork opens, this goes continuously, but in this case it has to wait for a long as it opens, as the replication fork opens, and then can synthesize another piece of the strand. This is the leading strand, okay, and this one is the larging strand. And these fragments are the Okazaki fragments. Okazaki, which is the surname of the person that discovered it. Okay? The Okazaki fragments. The fragments of the strand. Finally, when replication has taken place, a new enzyme is going to come here, which is the enzyme is called ligasa in Spanish, okay, and is going to join or to bond the different fragments, and the duplication of DNA is going to finish. So that's all about duplication or replication of DNA. Remember that it happens during the S phase of the cell cycle and before the cell division. On the other hand, we have the transcription and translation. Transcription and translation, the only purpose of these actions is making proteins. You know that when we eat proteins, okay, when we have proteins in our diet, we digest them and we break the bones into amino acids. Those amino acids go to the blood, okay, and the blood take them to the cells. And our cells are the ones which are going to pick those amino acids and synthesize their own proteins, our own proteins. How this occurs? Okay, the ribosomes are the ones that make the proteins or synthesize the proteins. So how do they know the order of the sequence of amino acids for making each different protein? Because that information is contained in the DNA. But DNA cannot get out of the nucleus, and ribosomes cannot get into the nucleus. So the DNA has to be transcribed into messenger RNA. And the messenger RNA, which is in green, is the one that is going to get out of the nucleus. The ribosome is going to read the RNA, the messenger RNA, and is going to put the different amino acids, methionine, proline, arginine, lysine, okay? These amino acids, which are in the cytoplasms, okay, are taken from the cytoplasms to the ribosomes by the transferent RNA, okay? It's a molecule of RNA. You can see that here is the 3' extreme and the 5' extreme. It's a single strand, okay? Which a single strand, okay? And, well, I'm going to explain after. And here are the bases, the nitrogenous bases. Okay. First step, transcription. As I said previously, the DNA is going to be read from the 3' extreme to the 5. So here the same. It is read by the 3 from the 3 extreme to the 5 extreme. And the new strand is going to be formed from 5 to 3. Okay? Here is the 3 extreme, so here is the 5, the complementary one. It's so easy. The difference here we have 2 strains, okay, and another 2 strains are going to be formed. But in this case, remember that RNA is a single strain molecule, so it's going to be formed a single strain. So this is the mold strain, and the RNA polymerase here occurs the same, okay, as here. Topoisomerase is going to uncoil the DNA. Helicase is going to open the fork, and the RNA polymerase is going to add the complementary bases here. So where is a T, it's going to be an A. And where is an A, it's going to be the complementary, which is T. But in this case, it's uracil, because remember that RNA hasn't got timing. It has uracil, okay? So TA, AU, CG, AU, okay? And here we have the RNA messenger molecule. The RNA messenger, is going to get out of the nucleus and goes to the ribosome. The ribosome reads the RNA reads the codons, okay? Codons, which are 3 bases. A codon is made of 3 bases. And these 3 bases, each one of the 3 bases, these are called codons, codify for an amino acid, okay? AUG codifies for an amino acid which is called methionine. This is the codon, and this, which is complementary for this one, is the anticodon. The anticodon is in the RNA transferrin, in the transferrin RNA. Okay, the anticodon. How do we know which amino acids are going to are the ones that correspond to these codons? Because we have a table with the genetic code. I'm going to show you the genetic code. This is the genetic code, okay? This table. Here you have the first base, second base, and the third base. And in our case, the first base, the codon is AUG. So we go here and we search for the first one, A, the second one, U. So A and U are in this column. So, and the third one was G. So here, it is here, AUG. This codon codifies for an amino acid which is called methionine. This other one codifies for another one amino acid which is called isoleucine. Leucine, phenylalanine, serine, tyrosine, okay? This is the genetic code and is universal. That is universal means that is the same for every single organism, okay? Those codons codifies for those amino acids. And also it is said that is degenerate. Se dice que es universal y degenerado. Degenerate means that several codons, as you can see here, codifies for the same amino acid. Or here, several codons, several different codons codifies for the same amino acid. So now I come back here. Watching the table, we can guess the amino acid sequence of the protein that is going to be formed. So the first one, AUG, codifies for methionine. So a transparent RNA which the anticodon is going to take the methionine and is going to take it from the cytoplasm and to the ribosome, and is going to add here methionine. Okay. Then the ribosome moves and reads this one, U. So U codifies for, I'm going to check it, obviously. U is phenylalanine, okay? So another transparent RNA is going to take the phenylalanine to the ribosomes, and here is going to be the anticodon of these ones. The anticodon of U is A, 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 okay? But the ones that codifies is the one that is in the RNA, on the messenger RNA, not the anticodon. The codons are the one that codifies, not the anticodon. So here is going to the ribosome. The transferrin RNA is going to take the phenylalanine to the ribosome, and the ribosome is going to add here the phenylalanine. Then it continues readings, and here is CUG. And CUG codifies for leucine. So the same, and it's going to put leucine here. Okay? And as the ribosome reads the RNA, it's going to add more and more amino acids in order to form the proteins, because you know that proteins are chains of amino acids. Be really, really, really careful with this. DNA is read from 3 to 5. From 3 to 5. And the new strand is going to form from 5 to 3. From 5 to 3. But RNA, the messenger RNA in the ribosomes is read from 5 to 3. And be careful with this, because if you do an exercise and you do it reading the RNA from 3 to 5, you are going to say G, U, G, U, C. And that codifies for a different amino acid, okay? The sequence is in that direction, from 5 to 3. C, U, G. And be careful, because the direction can be here the 5 and here the 3. So you should read in this direction. Be careful, from 5 to 3 in the case of RNA. RNA is read from 5 to 3, but DNA from 3 to 5. And so this is all. If you have any doubt, you know that you can ask.